43 people online, and it's just a couple minutes after 3. So I think I'll go ahead and start introducing our speaker for today, Dr. Charles Chen. And it's a pleasure to have somebody talk about something about besides corn and soybean in Iowa. So welcome and thank you for sharing your research with us today. Dr. Chen is a professor and peanut breeder at Auburn University. He got his PhD at the University of Illinois Urbana-Champaign, and then he served as a lead scientist in the National Peanut Research Lab in Dawson, Georgia, for 5 years before taking his present position at the University of Auburn in 2012, where he does genetics and breeding research in peanut. We'll turn it over and let Dr. Chen take it from there. And hopefully I had all those details correct in your introduction.
Okay. Thank you. Good afternoon, everyone. First, I would like to appreciate Dr. Yu's invitation. I believe this is a great honor to offer this seminar. The plant breeding and genetic program at the Iowa State University is very prestigious in the world. You know, when I did my PhD at the UIUC, I took my plant breeding class. We used Dr. Welder's very textbook called The Principle of Cultivar Development. So Dr. Freire is one of my most respected soybean breeder. I actually visited his soybean breeding plot at AMS in 2003. Currently, I'm working on the peanut breeding, but my training is on the soybean. So switch from the soybean to peanut is big change. It's from the ground to the underground, from the diploid species to tetraploid, and from the small seeds to large seeds. But one thing is not a change, which is the principle of the plant breeding. For today's talk, first, I would like to go through the peanut production. I believe you are all like me from Midwest with no idea about the peanut. The second part, I would like to talk about the genomic research, and finally go to my peanut breeding work. So here is the photo showing you the peanut, or we call the groundnut, is a self-pollinated species with indeterminate flowering. Is flowering above the ground by the set part under the ground that you can see through the peg. So that make a connection between the pod and the stem. And it is allotetraploid. Is 2x is 4, 2n equal 4x. It has a 20 pair of chromosome with the A genome and B genome. Is an oil crop with a 45 to 53% oil content and 22 to 27% of the protein. So cultivated peanut is origin from the South America. Here is from the Bolivia, Brazil, Argentina, and then they first introduced to the Africa, and then coming to the North America. And then another route is introduced to the India or China in the South or East Asia. So right now, peanut is adopted in the tropical and subtropical climate. So here is the like a top 10 peanut produced country in the world. The USA is in the third place with 4.1 million tons behind the China. They had 18.7 million tons and India. So after that, the 7 countries are Nigeria, Burma, Indonesia, Argentina, until Ghana. So this is like a USA peanut production by state in the every year is about 1.5 million acre. So basically you classify the 3 growing area. First one is a southeast area, including the Georgia, Alabama, Florida, and Mississippi. They count about 75% acreage. And second one is North Carolina, South Carolina, and Virginia. They are about like 15%. This area mainly growing Virginia. The southeast area growing the Groundnut type. The last area is in the southwest area, Texas, Oklahoma, and New Mexico. They growing probably about 10%. And the acreage there, believe it or not, Alabama, we are the second producer of the peanut in the nation. And this figure is very interesting. They are showing you the peanut yield increase over time. So you see the blue line here is acreage. The red line is the yield. So in the 1940, the acreage is reached to 5.2 million acres, but right now only like 1.5. But for the yield from the beginning of the last century, it's like 600 pounds per acre. Growing right now is on the average, we produce like 4,000 pounds per acre. So you count about the gains, the increase like 29.9 kilogram per hectare per year. I believe that the yield increase is mainly due to the genetic improvement. So the peanut is usually planted as a single row pattern with 36 to 38 inch spacing at the seed rate of the 6 per foot. Before the planting, the peanut seed usually change the fungicide to prevent the seedling fungus like a root rot disease. And this like the field preparation, some farmers, they prefer bedding, some don't. And these are most common irrigation system. You can see the picture here use a pivot, but you also can see a small number of the farmers, they also use the draped hole as well. And one very critical, also very unique things for the peanut farmer, also is a big challenge, is to determine the maturity date. That is very critical for harvesting. Unlike the soybean, you guys can see from above the ground, but for the peanut underground. So what we can do here is we call the whole scribe maturity profile. Usually like 120, 130 days after planting, the farmer go to the field, take the sample, and use this high pressure washer to blast the peanut pod. They will remove the X cap from that is the outer layer of the pod, and then you will see after remove the X cap, and then you can see inside of the mass cap. Based on the color change, you can determine the maturity. So, and then after you blast the peanut pod, and then you place those peanut on the board called the peanut maturity profile chart. So on this chart, you have a yellow line, yellow dark, orange in color, brown color, and black color. So on the majority, the peanut fall into the orange, black, and brown color, then it's time to pick the peanut. So for the harvest peanut, usually take 2 steps. The first one we call the invert the peanut. You can see the machine after invert, the peanut is upside down, and then let it dry on the ground for 4 to 6 days before going to pick a peanut. The main reason here is let the wine getting drier. Also, the moisture will reduce in the peanut parts. And then the peanut picker here will come in to pick it. At this time, the peanut moisture will blending down from 30 to 45% in the fresh to the 20%. So, and you see, you go to picker when the basket is full, you are pouring the peanut in the semi truck, and similar track will transport called the buy point here. So, and then those peanut in the similar track will load transfer to this dryer trailer. So after you put in there, those trailer will hook up with the air, hot air. We call this is a curing process. So we will bring in moisture down to the 8 to 10% for safe storage. And then before you put in the storage, you needed to take the sample here. So like I say, this is the trailer, and you take the sample and do the inspect, and then you can establish the grade, and then based on that grade, so you can to know how much premium paid to the peanut farmer. So, and this kind of different storage house, like a warehouse, like stone shape. This is like a conventional farm stock, and then you will be putting all the peanut there until the manufacturer called in the sharer, the product, they started to sharing. So the sharing, usually you can remove the soil and wine, and then you also go to like based on the size, you will classify it's a different the peanut. And then also here is called the sorting machine. Every single kernel will go through that machine. The machine can eliminate the discolor or defective kernel. So finally, you put it on the big bag here. That's the shared peanut ready shipping to the manufacturer to produce all different products here, like a soybean flour, peanut flour, peanut oil, peanut butter. So, and usually, unlike the other country where peanut are mainly used for the cooking oil, but in the US, we have 45% for the peanut butter, 21% for snack, and 15% for candy, and 6% in the consumer in-shell, only 4% as an ingredient. So now I hope you're all like eating peanut. So now let's talk to the peanut genetics. Okay, as we know, the peanut is a tetraploid. It has a genome and then also the B genome. So we also have a hypothesis that A genome is originally from the wild species Duraensis, and B genome is coming from the wild species Hypoensis for the B genome. So that's two wild species to make a cross, has the infetal hybrid. Somehow they had a chromosome double and founding the cultivated peanut. But that hypothesis is not get approved until the International Peanut Genomic Initiative group, they accomplished the whole genome sequencing of those two ancestral species. So in 2016, then published this paper, and on the base of the high DNA identity of the Hypoensis genome and then B genome, they also based on the biogeographic evidence, they conclude that the Hypoensis, they are be the descending of the B genome to the cultivated peanut. So here is like an example showing how cultivated peanut genome match into the two wild species genome. This photo is showing you is hypogaea, that is the cultivated peanut raised, how mapped to the Duraensis on the A5. You can see the blue dot, that is the presenting the percentage identity rate in the tilling path. So you can see that here is like based on the density, you know, in which segment of the chromosome here they are matched. Also, you can see the Hypoensis on the B5. So from this end to that end, you can see that always the blue, these are perfectly matched rate here. So another evidence that they are showing you here is the photo showing you these are 3 species, hypogaea, duranesis, and hypoenesis. They are occurrences in one single location. In here is on the Argentina, Paraguay, or this is a Peru in this at this area. So and after like 3 years, International Peanut Genomic Group, they also published the whole genome sequencing of the cultivated peanut. So in this paper, the result indicated cultivated peanut is the hybrid origin and contains essential completed set of chromosomes from 2 species. And then interestingly, the result also indicated cultivated peanut, they are evolved through the mobile element activity, deletion, and homologous recombination. So by another interesting here is as we know, in the wild species, we have another wild species called Montecala. Montecala actually is a tetraploid species. And also it's the only one species existing there. So the interesting question will to ask what the genetic relationship between the hypogaea and the Montecala. So here is the answer. So that in the 2020 cover featured paper here, there is an international group. They working on the Montecala. They do the whole genome sequencing. By the way, I also involve this group. So the group, they are sequencing the 17 wild diploid species. They contain the A genome, B genome, E genome, K genome, and C genome, and they compare to the cultivated peanut and the Montecala. So, and then they also finding that it's called a monophenotypic origin of the A and B genome. They are contributed the tetraploid species. Also very interesting, this paper, they also found the structure variation that is the contribute to the peanut part size, the increase from the small part size to big part size. So here is the showing you, so they are based on the subgenome origin and the phylogenetic analysis. They recommended they reconstructed a new evolutionary model from the wild and cultivated peanut. You can see that this photo here. So right now they are believe that like I said, duranesis, they closest with the hypoenesis, they formed in the Montecala. So Montecala name from there to domesticated to the cultivated peanut here. So for that, it's take about 10 years for International Peanut Genomic Initiative to complete the whole genome. But through the project, the good things for the peanut community, we build this called a peanut base, the database. I know in this database right now is a manager of base in the Iowa State University by Dr. Steve Connor. So it's not as good as a soy base, but I think this is a good achievement for the peanut community. So far we have 48K SNP chip can use by breeder. Also we have 489 QTLs for the breeding program. So let's move on. If you talk about the breeding program, the one thing is about the genetic diversity, we call the genetic resources. So this table showing you the major collection in the world, we totally around about 61,000 sections. So they had in the India is Arachis, also the India IBPTR. They have a bigger portion, but in the USDA collection, we have roughly about 10,000 sections. Also, we have maintaining some of the wild species. So, even we have 10,000 sections, looks like a big number. But if you look in the USA peanut pedigree here, you will find actually we have a very narrow genetic diversity in the peanut cultivars here. So basically, you can trace back all the cultivar is deriving from those like roughly about 15 PI or local race. So, but even you also can separate by this group. That group is mainly the Virginia type peanut. So another group, this group is mainly a Groundnut type peanut. If you consider the Virginia peanut, the genetic base even narrow. So, but also on the other hand, so you have a bigger number of the collection, but if you want to do some research, it's very tough to handle those bigger number. So in 1989, Brown, he introduced a concept called co-collection. That basically idea is trying to use a smaller collection to present the maximum genetic diversity. So I believe in the soybean community, they established the mini core or core collection. We are doing the same thing in the peanut community. Dr. Holly Cooper in 2005, he published paper, he established the mini core collection about 112 sections here. Also the India, China, they also do the similar things. So for that mini core collection, we still have a bigger genetic variation of about the seed and the pod. This photo just showing you what is the genetic variation about the pod size. You can see the different pod size, also the shape. You have a very narrow reticulation. Also, you see the pod size here, and also these are mini core covered 4 different botanical variety: hypogaea, vigorous, fasciata, also prevenia. So this one is like about the variation on the seed coat color, also seed coat pattern. You see this one has a bicolor, and also some like here the shape, the seed size, they are very different. So this geographic distribution of the mini core, they pretty much presenting from the different country in the world. So when I started doing my peanut breeding, the first things I request the mini core collection growing in the field. So the first thing I found is the heterozygous within this mini core collection. You can see in the field, I observe you have a variation. This one row suppose is a 1 PI, and you can see the leaf color. This is dark, this is light green, and then you also see the different stem pigmentation here. This one is a green, this one is a purple, and then also the vigor here. So also on the other hand, I see the genomic research has highlighted the need for the homogeneous genotype. So I decided to take a 3-year to work out about the purify those mini core collection, go through called the pure line process. So the year 1, so I planted the 20 seed, and then from that 20 seed, I select the 5 morphological uniform plant. And then second year, I planting those single plant as single row and take off the off type, and then also most homogeneous row, then we harvest the seed. And then third year, then you continue working on take off type, also get all the seed, homogeneous seed, and then for the seed increase. So, and then on the 2014, I registered those seed as a purified section for the US peanut minical collection. So after I do the purification, so this section not only preserve the similar genetic variation, but also becoming more homogeneous you compare to original section. After I deposit all the peanut section in the gene bank, so you can access through the NPGS. So after I publish this one, there are a lot of requests in the peanut community. So the utilization, like here, Dr. Dana in the NC State, he will use this one to the whole genome sequencing. And Dr. Kelly and Chamberlain, he was doing the screening for peanut smart in Argentina. And the Arachis scientists in India, they screening for heat strain. This is a drought in China. And the new assistant professor, Dr. Avalos Sanjay, he was screening for root structure. And Dr. Fadan from National Peanut Lab, they screen for the drought tolerance. And then also in my group, we did a lot of the G1s, and we're also screening for the leaf spot and then TSWA. So I can see this work is pretty straightforward. I get a lot of the impact in the peanut community. So let back because I clean up the minical collection, and very fortunately I applied the Peanut Research Foundation. I get a funding the project doing the GWAS to like association with the DNA marker with the desirable trait. So in that panel, I phenotyping the 120 genotype, including the 104 is minical and 6 elite line. And then I use that chips like a 40K, 48K SNP chips. The trait I do in the phenotyping, I list here is like an agronomy trait, mainly is a yield, seed size, a grade, and the disease evaluation for early leaf spot, late leaf spot, tomato spot, will virus, and white mold. And we also doing some seed chemistry, fatty acid composition, sugar, mineral concentration, flavoring, and risk water. And we're also doing like a flavor. Some of the seed chemistry, that was Dr. Minli Wang from USDA ARS. In his lab, he helped do all the analysis. And also Dr. Lisa Dean, also from the USDA, he did the flavor analysis. And by the, for the Peanut Research Foundation, they encourage the 4 priority for the peanut community to do the research. The number 1 is on the leaf spot and flavor, aflatoxin, and then drought. In my group, except the aflatoxin, I do all the 3 priority trait for the peanut. So today I probably will going to report some result for the leaf spot research and also the drought research. So this is we use that panel and we do the leaf spot the research. This paper published on the 2020 in the TAG. So basically, we do the phenotyping in our research station in the headland station. So we planting the panel in the field. It's easy to do the leaf spot evaluation. You just plant there. If you not apply the fungicide, I can say, I believe you will get the leaf spot there. So you can, this picture is showing you how it looks like. You can see in the here is a very susceptible line, and this line here looks a very resistant. So this like we do the early leaf spot, also we do the later leaf spot. This like is the frequency distribution is like a normal distribution in a different year. And then we do the from the 13, 14, 13, 14, and 15 here. So the disease rating is a very consistency from year to year. You can see this the correlation coefficient, and then we also just do the regression on the 2014 and 2015. You can see this linear regression with R square equal to 0.75. But also the meantime, we're also screening for the later leaf spot disease to see which line has the best performance. We identify this for PI has the highest resistance to the later leaf spot. They have a very low the score. Actually, the score here is we have the 1 to 10 scale. So everything below in 4, the plant pathology will consider that is the high resistant line. This 4 line coming from the China, coming from this country, I really don't know. And then also coming from the Sudan, Israel. And then we use the SNP chips. We do the GWAS. Here is showing you some of the genetic structure of the 120 genotypes. This is a kinship plot. So basically, you also can say, as I tell, in the mini collection, we have 4 botanical variety. So basically, they also cluster as the 4 groups. And this one is we call the LD decay. So you can see in the peanut, LD decay is around the 160 kb, and the R square equal to 0.2. Even that is a little bit lower, but I think that's good enough to pursue toward the GWAS study. So, and then we identify some of the like early leaf spot, the QTL here on the B9, on the A9, also the B9. This is a later leaf spot. We are interested in seeing is on the B9. We also finding exactly the same for the leaf spot as for the later leaf spot as for the early leaf spot. And then you see the notation for those the QTL is related with the TPG binding protein, and some you can see here is the NBARC domain disease resistant protein. So what we found in this study, you compare to other QTL, and we know, like I say, the B09, the QTL on there never ever reported before. And let way back to some of the research going on my drought study. So this is like a justification we're doing on peanut drought study. The approximately only 60% of peanut acreage is a non-irrigation land. Also, like I say, we have, even we are in the south, the drought prevailed in the recent decade. And then also, like a picture showing you here, if the drought occur in the early stage, they were devastating. And more importantly, it's related to with the aflatoxin here. This is the map showing you in the 2016 in the September. Is a later season drought, and that will create a lot of problem with the aflatoxin. So that's a possibly mechanism for drought stress, the drought tolerance. One is escape, but I don't think that apply to the peanut because peanut, we have a long growing season. Peanut maturity take about 130 days to 150 days. And we have some of the mechanism for the resistance, also the tolerance here. Then the moisture required in the peanut development stage, one is the germination. You need to have the water to have a stem established. You need to have in the flowering, they request a lot of the water, otherwise will cause the immunity part to abort. Also the feeding stage and also maturity stage is increase of a toxin contamination there. But we really wanted to ask a basic question is, when does the peanut plant they are sense the water stress and what is the critical stage and how they affect the peanut. So for that, so I studied this project back to 2009. So I first evaluation 24 genotype in this field, only ½ inch for the germination. And after that, only 2-inch rainfall for the entire growing season. So you can see the field is very, very devastating there. But fortunately, we also find called the C7016 is a very good drought tolerance. And then we use that one with adding another couple, totally of 5 genotypes. We do that in the rain or the shelter in the National Peanut Research Lab. So we have a treatment stage. We'll have an early season drought, middle season drought, and later season drought. And we are set up the drought treatment is like 4 weeks drought stress, and then you have the recover. And this the 2-year research from that result, you see there's a t-test table for the treatment from the irrigation, non-irrigation. So you can see the biggest impact in terms of the drought is 6 days after planting. And then you also see the t-test for the different genotype here. So we find C7060 has the best performance, AP3 has the poorest performance. And then we also looking for physiological response, like we call the relative water contents in the 5 testing stage. You can see the photo here in the middle season, and you can see the Georgia green. There's one genotype has a good performance. For the C7016 has a good performance in the later season here. So we are showing these 2 varieties maybe have a different mechanism here. Also, Dr. Faden in National Peanut Lab, he used a real-time PCR working on the A peanut transcription factor. Compare this AP3 and C7060, you can find the PCR, the gene expressed on the AHER-F1 has 12 times increase in the recovery stage. And then you also can see this ERFA also have a 30 times increase. So this the 2 papers we published there. For detail, you guys can go there. And then also, like I say, we are use that mini code collection. So we are screening for the more drought peanut tolerance genotype. So you can see this is the yield after drought. You can find these 2 lines has the best performance, and these 2 also pretty good. And this one you can see is poor. Then what is the differences drought tolerance mechanism here? We have a Dr. Avalo Sant. He's a plant physiologist. He did a photosynthesis. You can see these 2 best performance line here. So they have a highest photosynthesis. That means they can get a lot of the water to do the photosynthesis. At the meantime, you also see this tool, they have much less the photosynthesis, but they still maintain a high yield. So that give us a clue. This genotype may involve different mechanism of the drought tolerance. So the Dr. Sanjay, he was doing all different the primate over there. So later on, probably another year, we can have more result. So another job here, work here is we're also doing some of the iron-seeker analysis here. So we have the use for genotype C7016, also the T-Groundnut here, and then we also these 587 and 506. That is the IL line derived from the C76 crosses with the T-Groundnut here. We do the we do the drought treatment, also the non-drought treatment. So you can see we finding the final finding, we finding the 43 KEGG pathway involved to the metallic pathway biosynthesis of secondary tolerability. They are all involved the drought tolerance. The figure is just to compare drought tolerance and drought susceptible here. This one is expression qualified ABA related were like ABA related DGE. And finally, I will move to my breeding program. So this is like a breeding goal. We need to set up is like you need to satisfy the different consumer like a farmer. We are breeding for the yield, disease resistant, drought tolerance, and they were asking like a uniform size, easy to share and manufacture. They are more like a higher lay, also is a low aflatoxin. Finally, the consumer like you and me, we are more like to have a health needs and then flavor. So compared to the soybean breeder, I think a peanut breeder, they have more consideration because you have more customer here. So this like my philosophy about how your integration of the genomic transgenic technology in the peanut. You can see the figure here. So you can get the genetic diversity from cultivated peanut, also wild relative, and you go to the conventional breeding through the pedigree or backclothing. And then we're also conducting the genomic research. You find in the tree, the mark association to the marker seed selection. You can have a gene discovery, and then you go through the gene editing or biotech, get the GM peanut. But so far, the peanut industry, they don't like a GM peanut because the peanut mainly consumer as a food. So, but anyway, my philosophy is we need to integrate all the components as the final goal is to release the variety. As a breeder, this code is I like it. This code as a plant breeder, saying plant breeder, I integrate all the information from the many disciplines, including the breeding methodology, genetics, genomics, statistics, economy, pathology, economic, and profit. I like to turn here the breeder where the prophecy. So this like a variety I just a couple years I released is a runner type of peanut. So this runner type of peanut is the high yield and medium maturity, very resistant to TSWA and leaf spot, and then also has a higher lay and very good flavor. This the pedigree is I make a crosses in the 2008, use the York and then Tifka and go to the breeding selection. And then also, like I say, during the selection process, I selection for the leaf spot. These are selection for the higher lay. I use some of the marker for the like leaf spot resistant. And then in the 2005 14, 16, 14, 15, 16, I conducted a yield trial and finally released on the 2017. So this like some of the yield comparison, yield performance. These are the 3-year, and these are the 2-year in 3 states, Georgia, Florida, Mississippi here. So you can see the AU17 compared to G060, which is the most popular variety nowadays the farmer grows in these 3 2-year location, 2-year in 3 location. They are all higher than G060. These are the year performance in the North Carolina and Alabama. So you can see that. And these are disease resistant for disease rating for the TSWA leaf spot. So the AU17 is on the top. The smaller number are better resistant. So then here you can see the real in the field performance. Here is the AU17. You can see some of the TUF runner 150. You can see they are all gone here. So that's a good sign. And also in 2018, the one farmer grew the AU17 in his farm for the largest increase in the 100 acre. So average yield is made for about 6,245 pounds with the 73 totally sound maturity kernel. So last year, Alabama farmer, they grow this peanut a lot, roughly about 10,000 acres because the seed supply we don't have. So I hope that this year the acreage will goes up. So I almost done that. Besides, I presenting here, these are some publication on the other tree from our lab, like here we're also doing some of the medical variation trait for the QTL or GWAS. We also do the growth habit related trait. And we also collaboration with Dr. He from Tuskegee doing the CRISPR-Cas9. And the Chinese group, the evaluation for the drought. And this one also, there is one student, Jin Lee. She is a PhD student now in the agronomy department at the Iowa State University. She conducting the TSWA, this publication. And finally, I would like to acknowledge the funding source from the Peanut Research Foundation, from Peanut Producer, Alabama Peanut Producer Association, and Peanut Board, American Peanut Sugar Association, NIFA, also the ARS. And this is my collaborators from the Auburn University, has Alova, Dr. Alova, Sanjay, Dr. Fang, and also Dr. Yuan Bao. I know Yuan Bao, Dr. Yuan Bao, he got a PhD from Iowa State University. Now he is the assistant professor in the biosystem department in Auburn. And I have a lot of people from the USDA AIS collaboration, Dr. Minli Wang from the AIS, Hua Deng, Lisa Ding, Chris Batts, Marshall Lam, Colin Holberg, and then also I have the collaborator from the Hansen Alpha, Tuskegee, University of Georgia, NC State, also from China, Hebei Agricultural University in China. These like some of the former students, some of the new students, and these are my technicians from the peanut lab. So we are working together in the field. So for that, I will finish there. I thank you for your attention and open for the question. And I also welcome you guys have the chance to reach the Auburn University. Auburn University is very beautiful campus. Seeing is a believing. Thank you.
Okay, thank you, Dr. Chen. All right, let's, as Dr. Chen said, we'll open up for questions.
Yeah, Dr. Chen, that was a great talk and really appreciate your time today. I was really intrigued by your the graph showing the yield versus the supply or the acreage that's being grown in peanuts in the United States.
Yeah.
And it looks as though even though yield's going up, acreage is staying about the same. So are you seeing your local commodity boards trying to create more demand for the peanuts, be it through different processes outside of food? Like in soybeans, we're seeing some of our soybeans going into asphalt now. So I'm curious, what are other uses that are being driven for peanut yield?
Okay, you mean the peanut yield or peanut acreage? You mean the peanut acreage, right?
Well, we're seeing, it looked like your acreage was staying about the same or even going down, but the yield was going up. So what's driving that increased usage of peanuts then?
Actually, in the USA, peanut production mainly consumer by like a consumer in the USA, but we do have some of the export. So mainly it's go to a European country. But I think the limitation for expanding the peanut, number one, is as I talked before, it's not like I say the farmer want to grow the peanut, they can grow it because the main challenge is after you harvest the peanut, like a post harvest, you need to have the buy point. So and then you need to have the storage, you need to have the shearing plant. So like not only the peanut, the farmer want to grow the peanut, you need to have other facility to match up. So that is a kind of the difficult if you not see the big profit, So I think, but I do see the peanut growing acreage is going to up because like I say, whenever the China wanted to buy the more peanut, and then will goes up.
Okay.
Yeah.
So do you think that the current market is able to move the demand for peanuts, or does there a lot more investment in infrastructure need to occur for the extra demand?
Yes, I think the infrastructure need to occur before we can move up.
Do you think the infrastructure will keep up with your breeding efforts then?
Yeah, but like I say, the Mississippi State, for example, like 10 years ago, they don't have a lot of peanut. Right now, they increased like 24,000 acres. I also heard that Arizona, they also started to grow in the peanut. So I think if once you have some acreage, the industry will build a buy point there.
Yeah. Awesome, thank you.
Okay, are there any other questions? Oh, Stephen.
Yeah. Very nice presentation, Charles.
Yeah. Hey, Stephen, nice to see you.
Good to see you. So the peanut research community has found that a lot of the diversity in cultivated peanut has arisen through subgenomic changes.
Yes.
Which is different than a lot of other crops. Do you ever encounter what you think is a pure line where a new rogue appears? In other words, do you think you see new diversity arising?
That's a very good question, Stephen. So actually, couple of, like I say, I still growing those mini core like almost every year. So I do see some of the variation. I can still observe some variation there. But you compare to original seed I got from the gene bank, now the pure line, I think is get much improved. Yeah. So I think if you are doing some of the like a SNP doing the genotyping, you can see some variation in there.
We've certainly seen variation within accessions in the core and the mini core. So purifying is really important.
Yes. Yeah.
Hi, Dr. Chen. Good talk.
Yeah.
So I was just wondering, you showed how peanut was spreading from the center of origin to Africa, to India, to China, and you assemble this minute core and you evaluate the drought tolerance, and you a couple of lines or more are showing its superior drought resistance. So where those lines are coming from? I was just curious about that.
Oh, sorry about that. I don't have information on hand.
Okay.
I can
Okay.
send you email. If you're interested.
Okay.
Yeah, I'll send you an email. Sorry about that.
Okay. I was just curious whether they're coming from, because I guess India or southern China.
Okay.
In
sure,
southern
sure.
China, maybe drought is not a problem, but in Africa, maybe that.
I see.
Yeah. I was also wondering whether anyone artificially created hybrid based on the putative progenitors.
Oh, actually, in the peanut community, there's a group in the UGA. They also working on the synthetic tetraploid. They make the crosses with the duogenesis and the hypogenesis and then double chromosome. If you want to integrate of the desirable gene like the leaf spot resistant gene, because the diploid you're not able to directly make crosses with tetraploid, you need to make a synthetic tetraploid, and then you make the crosses with the cultivated peanut. They did that way to integration some of the desirable traits.
Okay. We can
I
have
have a question.
questions, but I just want to take this have this opportunity for everybody to thank Dr. Chen again for his seminar. Real applause or applause emojis.
Yeah,
If
thank you.
4 o'clock is the normal ending time, but we can continue and have discussion for as long as people want to stay on. So
Yeah.
hopefully, Dr. Chen, you're willing to stay with us and answer a few more questions and engage in discussion. So
Okay, sure.
thank you. All right.
Okay, yeah, thank you.
More questions. And one comment while we're waiting for somebody to ask a question, we're up to 56 participants today. So that is really wonderful to see that many people plug in to watch your seminar today. So
I have a question, Doctor. Like, how's the mutation breeding affected to the peanut production? Because I am originally from Sri Lanka, but I live in Saskatchewan, Canada.
Yeah.
In Sri Lanka, they are growing one variety, I think, developed from the gamma irradiation. I think they are growing for 25 years. It is the 80% of the cultivation amount in Sri Lanka. So how's the mutation breeding affect to the peanut production?
You mean the mutation methodology for the peanut breeding?
Yeah.
As far as I know, in the US, we don't do a mutation for the breeding, but the Dr. Peggy Ozaya in his group, they doing some of the tilling work for the peanut. They mainly is working on some genomic research. But I do know the Chinese peanut breeding program over there, they did a lot of mutation for the breeding.
Hi, Dr. Chen. I had a question for you. Great presentation, by the way.
Yeah.
You're running a lot of these GWAS, finding QTL regions for diverse traits. What's your strategy for integrating that diversity into your breeding program? Are you running a lot of like three-way crosses, extensive back crossing? What's your strategy for bringing in that diversity to your program?
I think that's a good question. So I collaboration with a scientist from the Hansen Alpha. So right now we want to use for the genomic selection. So mainly, I will provide in my elite line if the yield performance is good. For example, I have a line called the AU 16.28. The yield performance very good, but they don't have a good resistance to leaf spot. So I make crosses. We know in the field line, we screening from the PI, and then from there, they doing the genomic selection. They are working on the growth chamber. That growth chamber, we can make 3 generations a year. So they just doing the back crossing. So you are going to use the genomic like a genomic selection for the background, also from the desirable trait, the gene. So that work we just starting that, but I don't know what's the result. Maybe we're waiting like 3-4 years. I can know that it's very successful. Also, I concerned what is the efficiency there, something like that. So basically, I think you have that genomic research is good for the back crossing. That's my opinion. If you want to do the back crossing breeding, that's very good.
Thank you. Appreciate that.
Yeah.
Hi, Dr. Chen. It's good to hear your talk. I'm a student in Dr. Cannon's group.
Yeah.
The Minicore and the core is a really great resource for the peanut breeding community. I've used the Minicore a bit in GWAS, and for that, some of our main findings were that a lot of difficulties just using the GWAS approach, especially in the Minicore. And the biggest reasons for that we found were just a small collection size, and then a lot of similarity between the accessions, although they were selected to be very diverse. What are some of the challenges you've, I guess, come across for the traits that you focused on? Thanks.
As I said on my presentation, I focus on the disease resistance. Now it's on the drought tolerance. And you also mentioned about the size of the Minicore collection. I completely agree with you that the size is smaller. Currently, I collaboration with Dr. Millie Wang from the USDA. He is the work with the curator of the peanut collection. Right now, we want to sample some of the lines. We want to expanding to about 300 sections. So actually, we are working on that. So it's from the whole 9,000 sections. So, and then I think that we call it a new genetic diversity panel will be a better presenting the whole genetic diversity. For example, the Minicore currently we have only covered 4 botanical variety. Actually, in the peanut collection, we should have 6 botanical variety. Another thing is Minicore, we don't have a high oleic tree. Now we are going to cover that.
That's great. Thanks for that. Maybe another question that I could ask is from the, you mentioned sort of like a narrow genetic base for the cultivars that are released here in the US. But we do have a lot of phenotypic diversity. You know, what are some of the things that I guess can explain that? A narrow genetic base, but a lot of phenotypic diversity.
Okay, you talk about but I can still phenotypic diversity. If you compare to the collection, I can tell on the release variety is much less genetic diversity. For example, the seed color, we all see the release the variety all the pink, almost only one color. Because like I say, the purple color is not a desirable color for make the peanut butter. Because if you have a purple color, the seed core, the peanut butter color will is not good, because the peanut butter consumer will like the peanut butter over there. So I don't know why do the breeding selection, all the seed is a purple seed, I threw it away. So that's my personal opinion as a breeder. I see the morphological trait in the release variety is very less diversity. If I do the evaluation for the mini core, I see a much more phenotype over there.
Thank you so much. Again, I appreciate your talk.
Yeah.
Dr. Chen, I have I'm sorry, someone is there.
Yeah.
Oh, okay. So I grew up eating a lot of peanuts. I grew up in southern China, so my family used to grow a peanut as well. But I don't know really much about different type. You talked about the runners type, and looks like a geographic regions in the U.S. in terms of production areas. Virginia and the Carolinas versus the one that in the South. What? And you also mentioned in your mini cores there are 4 botanical varieties. I'm a little bit confused about the different types of peanut and yeah. So.
Okay, I should do a more explanation about the the cluster of the peanut. So let us say that is for the species. So hypogaea is a species, but there's 2 subspecies called the hypogaea and fasciculata. So and then on the hypogaea, you have 2 botanical variety called the hypogaea, and then it's called hirsuta. Okay, and then for the fasciculata, you have 4 different botanical variety. So they call the fasciculata, Pervinia vigorous, and then Alvinia. So that is based on the botanical to classify. But we also say another way to talk about the peanut is a Groundnut type peanut, Virginia type peanut. Actually, Groundnut type peanut and Virginia peanut, they are the same in the hypogaea. So only distinguish is the part size. Like a small part size is growing mainly for peanut butter, we call the Groundnut type. And then big size is we mainly consume as an in-shell peanut or roast peanut. We call the Virginia. But on the genetically, you can easily make a cross. In my breeding program, I clone both the Virginia peanut and Groundnut type. So that is a distinguished. But on the fasciculata, you also sometimes can say that another term called the Spanish, also the Valencia. That's on the other species. Yeah, but the hypogaea and fasciculata, even they are the subspecies, in terms of the crosses, there's no any barrier over there. You can easily crosses. Yeah.
Hi, Professor Chen. Thank you for your nice talk. So I have a simple question. When you develop your pure line, what are your basic major consideration to choose the cross, the parents, to create the create a population to develop the pure line?
You mean developing the pure line? You mean the breeding for the breeding? Because
I assume you you choose different variety or different parents to cross, and then to select pure line from them. Is that right?
yeah, okay. So for the breeding process, because the peanut is a self-pollinated crop, only less than 1% of the crossing. So in my breeding program, I use called a pedigree selection. So what that means is I choose the two parents, make a crosses from the F1, and I go in the F1 to F2, I started to make a selection. So that in each generation, I pick the desirable plant. I go in as a single plant row. So within the single plant row, I keep a select until F5, F6. So and then in the F5 or F6, they are pretty pure. So then I doing the bulk selection and then putting in the yield trial. So when I put my peanut in the yield trial, basically, presumably there is the pure line.
Thank you.
Yeah.
When you start your the new population for the pure line, let's say yield the target trait.
Yeah.
So you just select two high yielding parents to make cross as the starting point of the F6.
So actually, that's how you to make the parent crosses. I think that's based on the breeder experience. So also, like I say, depends on what your objective. Like I say, you want to breed in a high yield. Normally, you will make a crosses with the high yield by high yield, but you also need to see what is the something you want to improve. Like I say, improve the leaf spot disease resistance. Then you need to take one of the parents has the high leaf spot disease resistance over there, and then you go there from the do the selection. So I cannot only use one or two sentences to describe how you make how you select the parents. That's really depends on what your experience. I do find some parents is we call the good parents, because from the progeny I can see the performance. They always has a high yield or some of the good performance. We call that is probably, I believe that is due to have a good general recombinability. Even peanut is not like a corn. They were working on called the special like a recombinability. But I do believe some of the parents there have the some of the way to show it has a higher general recombinability. So if you check the pedigree, you find for example, I know in C99R, that's the variety released by the UF, University of Florida. Right now you can see at least I know it is 4 or 5 new variety released from that C99R as a parent. So I do believe that C99R is a good parent. Yeah.
Okay, thank you, Professor Chen.
Mm-hmm.
So that would be a parent that would have a very good breeding value.
Yes, yeah.
Do you also see lines that have are maybe high performers but don't have good breeding lines? And I guess in other words, do you think there's a lot of epistasis and or other forms of non-additive gene action in peanut, even though it's self-pollinated.
Yeah. Yeah, I do see, for example, currently there's a called a Georgia 06G. That variety, actually, one of the parents is derived from the C99R. My line, AU17, has one parent. Also, you can trace back, has some of the portion of the genetic base from the C99R. And I make a lot of courses with a G06G. I can tell the progeny from that parent is a poor performance. Yeah, but I really don't know what's the reason cause for that. That will be a very interesting.
Hello, Charles. I
Yeah.
noticed that when you were in the earlier slide, you indicate we believe that peanut was originated in Bolivia, Brazil, Argentina.
Yeah.
Later on, there was a paper of sequencing those tetraploid or also those diploid parental species. And then on that map, you showed Argentina, Paraguay, and Peru. So
Yeah.
I think that by sequencing analysis so far, can we update the general regions? Or you think the earlier declaration of the Bolivia, Brazil, Argentina are still generally correct?
I think, I think that statement is still good because I still believe that domestication is not only occur in the one very specific area. Maybe it's abroad in another area. So, for example, like I say, the rice, I read some paper on the rice domestication. They are not saying only the one location, especially like you have a different subspecies like fasciata hypogaea. So if that's only one location, how you come out that, it's kind of really a complicated question. We need looking for more evidence to make that statement.
Okay, thank you. You also showed the map of the peanut production map. It's mainly in the south. What is the reason people talked about that why we did not push peanut to adapt to a little bit further north? Was because of we have other crops that have higher priority, or was that because of other reasons? You mentioned about the facility production.
Yeah, but
Another can push up. Yeah.
Another reason I believe is soil type. Peanut is like a grow in a sandy soil, and also like say they need a more temperature, like accumulated temperature point. It's need a bit higher. So that also the growing, the maturity count for 130 days, at least for 150 days. You needed to have a consistently high temperature.
Okay, so if we show you did show the production bar chart, you did not show the map in China, which is number one producer. But if you do that, it's still mainly in the south where you have enough.
No, that's a good question. Like in China, I know they grow the peanut as far north as if you're looking for the altitude, it's similar over Chicago area. But they grow in a different type of the peanut. It's called a fasciata, called the Spanish. The maturity date for that peanut can be as short as 95 days.
Okay. That's
but
good.
the yield is much lower.
Okay.
Yeah, much lower.
Give up something. Yeah.
Yeah. Mm-hmm.
Good.
So freeze intolerance is not an issue. it's just more of a degree days and the yield, those kind of things.
I heard my colleague in the north part of China, the breeder, right now they are working on cold tolerance. Because the problem for them is when they are planting the peanut in the early spring, they have a bigger impact for the seed germination under the low temperature. So they are screening for like a seedling and cold resistant.
I see.
Hello, Dr. Chen.
Yeah.
Thank you for the talk. My question is with regards to, I saw pictures you showed for drought breeding. We showed above ground material, tolerant and non-tolerant. And I just wanted to ask how correlated is the yield under drought with the above ground biomass?
Above ground biomass.
Yes, compared, like there's a lot of vegetation above ground. Does it necessarily relate to a higher yield in terms of?
Actually, we have one master student. Actually, she collected the data about the yield of the pods, also the biomass. Right now, I don't have definitely the answer, but I do believe, based on my observation, I find some of the peanut in terms of yield is not necessary. You have a bigger biomass about the ground, have the high yield. So I do see some genotype, the yield, like I say, the above ground, the biomass looks not good, but they do have a lot of peanut underground.
No, thank you. And last question is, you mentioned a lot of disease. Are you having issues with the aflatoxin issues? I mean, pre-harvest in terms of your problem?
Yeah, aflatoxin, I think that's a big challenge issue, not only in the peanut, but also in the other crop like corn, wheat, another nuts. I know the touch that question is, the bigger the challenge for breeder. So far, I don't see any genotype, either the tolerance or resistant to the aflatoxin there. The one thing is it's very difficult to do the evaluation in the field for the aflatoxin. Like I see in the field, if you can find one spot in the field has the aflatoxin, you will ruin your whole field product. And because from genetic standpoint, if you don't have the resistant, how are we going to breed in the peanut? So that's why I know the touch for that trait. But I know there's a lot of the effort going on to trying to find it. Yeah, but I have much more priority stuff in my breeding program I need to work with. So I just not touch that problem. Yeah.
Thank you very much.
Okay.
Hi, Dr. Chen.
Yes.
Great presentation. I have a good question. I have a quick question about from the breeder side. What is the critical yield component for you to improve the peanut yield in the field? The yield component, like pod size, number of the pod underground, or the kernel size or kernel weight, something like that.
Based on my breeding experience, I do see the one component is the peanut seed size. Like I say, currently the Groundnut type, the seed size is about like about 55 gram per 100 seed, and that is called a Groundnut type. If you go into Virginia type, it's like a 90 or 95 above. And if you are looking for the early on, they released the variety like a Georgia Green, Flavor, Groundnut. Those are medium size. Seed size is like 100 seed, about the less than 60 gram per 100 seed. But if you move into right now the new release variety like Georgia 06G, Florida 07, also my line AU17, those are present a high yield. But if you're looking for the 100 seed size, it's about 72, 75. So my impression is the seed size improved, the yield is getting improved. So right now, when I'm doing my breeding program in terms of yield component, I really pay attention to the seed size. At least that I feel like it's work for my peanut crop. Yeah.
Okay, thank you. Nice answer from the field breeder side.
Yeah.
Another question, Charles. Well, you were actually showing the pedigree breeding for your particular variety. It's called AU NPL 17.
Yes.
So this is, we have AABB genome, but it behaves as a diploid, right?
AABB genome. So actually,
but your heritage pattern is diploid, right?
Yeah. Yes. So
okay.
Behavior is pretty like a diploid. Actually, from the breeding standpoint, I don't see that's a big difference between the diploid or tetraploid. So as a breeder, you want to improve the trait. You are working on that. So
the complication will show up if you want to get out of the cultivated species for diversity. You want to reach to the AABB. Then, like you said, you have some, bridges you have to build, right?
Yes. Yeah.
Okay.
Yeah.
Well, that's clear because I wasn't sure. I saw you
yes.
guys have a lot of problems. Okay.
Yeah.
Good to know. Can you actually talk about, this is different from other, you talk about the grade, which you have a symbol SMK, LSK, and 6464. Type of a grading system, general, what is the general concept behind that?
Behind that is like I say, that is very important for the farmer, how you receive the premium. So because the shearer, they buy the peanut, they are going to share how many percent of the kernel they can share. That's called a sun maturity kernel. Sun maturity kernel, that means the kernel after sharing, they can sell to the manufacturer. So because the shell, some peanut has a very thick shell, so the kernel percentage is lower, like below 70%. That we consider is lower. If you have the shell only like only a 25% shell, then you can have 75% kernel, so you can sell 75%. But when the farmer sell the peanut to the buy point, is they going to weigh how many tons of the peanut in shell.
Okay.
So they were going to do the grading right away to see you want to treat the peanut, what is the percentage of the some mitotic kernel for that peanut? So based on that peanut, they were doing like a calculation, how much I will pay you. So when you sell the peanut, you dry before you put the storage, they needed to do the inspect for that. Also, I know there's a genetic control. I mean, some mitotic kernel that trait is a genetic controlled trait. Though some variety you have a high some mitotic kernel, some have a low mitotic kernel. That's also very important for the breeder to breed in that trait. So it's not like a soybean, the farmer, they will harvest the soybean. So they send in the soybean to sell them. So you know the weight. So.
Good, thank you. There are people still around. So what is your plan to start testing or start implementing or with genomic selection so far?
Actually, I got some funding. So last year, we make a lot of clauses. So actually, right now we are working on that project. Okay. My collaboration with Hansen Alpha. They were mainly doing the genomic, do the genotyping. Yes. So we are going to do the clauses and then do the field trial. Yeah.
Good. Thank you.
So I do need hire a new student working on that genomic selection. If you have a good one, recommend it. Yeah.
Dr. Chen, I have a question.
Yes.
How's the double haploid development technique has been used so far for the peanut breeding?
Actually, they are very commonly used for that. You want the integration, the desirable trait from the wild species. So, but in my group, I don't do that. And by the National Peanut Research Lab, down to the Dawson, Georgia, there's one scientist. They lately hired one scientist just working on that job. So just the integration from there, they call it a per breeding program. So once they enhance that breeding material and turn into the breeder. So, but I do know it's very, very successful to double the chromosome. Yeah.
Thank you.
Yeah.
Well, if there are no more burning questions,
yeah,
maybe we can wrap up pretty soon. Jinming, how long do we typically continue these discussions?
This is good, unless somebody just turn on the video, want to ask a question. No. I typically for good discussion, we want encourage people to turn on their video if possible, and speak a round of applause before we leave.
Okay.
If you can turn on your video, please. Okay, it's more fun, right? Yeah.
Yeah. Thank you.
Thank you, Jody. Thank you, Charles.
Yeah.
Thank you.
Yeah.
Jen.
Okay.
Thank you.
Thank you. Appreciate. Okay. Appreciate your time to listen.
Yeah. Wonderful.
Okay. Keep in
Bye-bye.
touch.
Yeah.
Yeah. Bye.
Thanks, Dr. Chen.
Yeah.
Thank you, Dr. Chen.
Thank you. Thank you.